Hello everyone, welcome to RG Education. I'm Dr. Vandana Kushaha. Today I'm going to discuss about BT cotton crop, which is a very important topic for all the net exams because it is a transgenic cotton crop and transgenic covered in the different types of the exams like SRF net, GATE Excel, GATE BT, T for exams as well as the different types of the agriculture exams. So it's a very important topic. BT cotton is a cash crop, I think you know, and it is a fiber crop which is used in the textile industry. Very basic crop. So why it is developed VT means what is the basic difference in the normal cotton crop and BT cotton crop. So BT cotton crop is has been introduced BT gene, that is Bacillus thuringiensis bacteria, means that gene is isolated from the bacteria which is introduced into the cotton crop. So it is going to be resistant against the different types of the insects like lepidopteran and cotton bollworms, American bollworm, different types of the bollworms. But in that normal cotton crop, what happens? When the insect attack on that, then it is going to reduce the yield of the crop because it is going to damage. It will damage the crop. Ultimately, whatever optimum yield will be there, then it will be reduced. But BT cotton have the resistance to the insect. So there are a lot of the chances to increase or improvement in the crop. Okay, so how it can be prepared means what is the technology we are going to discuss here one by one. Okay, this gene enables the plant to produce protein toxic to bollworms, one of the most destructive cotton pests. BT cotton was introduced in India 2002. You have to remember that is cotton bollworms is the main pest which is going to attack on the normal BT crop. BT cotton was introduced in India in 2002 quickly and became widespread. Means when it was introduced in the 2002. Today it covers the majority of the cotton growing areas, contributing to improved farmers' income and reduce the pesticide related health risk. So who invented means how it can be produced initially. So this is the basic important line I can say because so many in exams it was asked which company had produced BT cotton, which company has produced flavor saver tomato, which company has produced even golden rice. So these are the questions which are continuously coming in the different types of the agriculture exams. So you have to remember that Monsanto scientist means Monsanto is the company basically. Scientists inserted a toxin gene from the bacterium Bacillus thuringiensis into the cotton plants to create a caterpillar resistance variety. A joint venture between the Monsanto and Myco. If option will be there for the both, then you can take. Otherwise, main company was there, that is Monsanto. Introduced BT cotton in India in 2002. You can see here diagram that is larva feeding the cotton crop. Means it is damaging the crop. I can say that it is damaging the bollworm. Means boll. What is the mechanism basically? The insect disease causing organism Bacillus thuringiensis is naturally occurring soil-borne bacterium found worldwide. Means Bacillus thuringiensis bacteria, simply we have to first of all we have to isolate the genes from the Bacillus thuringiensis, that is a cryzine. CRY, that is cryzine. A unique feature is production of the crystal-like proteins that is selectively kill the specific groups of the insect and other organisms. Because cryzine have the quality to kill the insects and as well as the other organisms also. When the insect lepidopteran eats, yes यहाँ पे lepidopteran भी हो सकता है and it can be coleoptera. There are the lots of the varieties of the insects basically. If you will search in the Google, there are the list of varieties of given. Eats these cry proteins, its own digestive enzymes activate the toxic form of the protein. Cry proteins bind to the specific receptors. What it does basically, When you will isolate you will isolate the cryzines from the bacteria. Then after that you have to gene clone. You have to clone the gene. Basically, after cloning, you will be produce the lots of the multiple copies. Then you are going to use the vector that is called plasmid vectors vehicles, or we can use the expression vectors. Then you will produce the protein, means it will, that gene will give the expressions. Then you will produce the proteins. It can be produced into the plant only, because you are going to transfer. It is a transgenic Vt cotton crop. So you are going to all your vectors agrobacterium transformation के through आप वहाँ पे introduce करेंगे. Then that cryzine will be expressed as a protein, cryprotein. Its own digestive enzymes activate the toxic form of the protein. See, cryproteins bind to the specific receptors on the intestinal walls and rupture the midgut cells. Then insect will bite the plant, then it will introduce into that protein will introduce into that, and it will create the ultimately toxicity into the insect. So insect will die within 2 to 3 days. Okay, so you can understand better here by the diagram. First of all, what you will do? First of all, you will isolate the gene, that is cryzine, from the bacterium. Then after that, what you will do? You will use the gene cloning methods and you will introduce your gene into the plant. After you will introduce into the gene, it will express as a protein, cryprotein, that is called crystal protein. This crystal protein has the qualities of the pH. See, I'm not talking about crystal protein pH, because everyone's pH means, if we are human beings, we have the metabolic activity in our body as well as the different types of the cells of the body. Different cells of the body require different pH. That's why we have the blood like pH 7.4 or 7.2, and in the stomach acidity means, very acidity means 5, around 5 it is. So it's very important to work the cells or to work the anybody pH is very important factor. So what happens here, when जब ये क्रिस्टल प्रोटीन आपकी let understand, जब ये क्रिस्टल प्रोटीन आपकी किस में होती है? बैक्टीरिया में. कोई भी टॉक्सिसिटी कॉज नहीं करती है. क्यों? बिकॉज बैक्टीरिया का जो पीएच होता है साइटोप्लाज्म का, वो कैसा होता है? एसिडिक होता है. बट जैसे ही यहाँ पे आपने प्लांट में इंट्रोड्यूस किया, प्लांट में भी कोई प्रॉब्लम नहीं है, लेकिन ये प्लांट में प्रोटीन के फॉर्म में एक्सप्रेस होती है. ठीक है. अब जब इंसेक्ट इसको बाइट करेगा, खाएगा, तो जो इंसेक्ट का गट है, गट एक पार्ट होता है किसका? स्टोमक का, इंसेक्ट के, मिड गट, हाइंड गट, ऐसे करके. तो जैसे ही वहां पे जाएगा, उस गट का पीएच होता है एल्कलाइन. जब पीएच एल्कलाइन होता है, उस पीएच की वजह से ये जो प्रोटीन होता है पहले तो इनटॉक्सिक होता है, मींस टॉक्सिक नहीं होता, नॉन टॉक्सिक होता है. जैसे ही वहां पे जाएगा इंसेक्ट गट पे, ये टॉक्सिसिटी क्रिएट करता है, क्योंकि ये प्रोटॉक्सिन से टॉक्सिन में कन्वर्ट हो जाता है. मींस इनएक्टिव टॉक्सिन से एक्टिव टॉक्सिन में कन्वर्ट हो जाता है. जब ये एक्टिव टॉक्सिन में कन्वर्ट होगा, ऑब्वियस सी बात है, टॉक्सिसिटी क्रिएट करेगा. क्या करेगा? टॉक्सिसिटी क्रिएट कैसे करता है? सी, दिस इज द सेकेंड स्टेप, लार्वा मिडगट प्रोटियोलाइटिक डाइजेशन. मीन्स सब कुछ ऑन द स्पॉट नहीं होता कि इंसेक्ट को टॉक्सिक पहुंचा और इंसेक्ट खत्म, ऐसा कुछ नहीं होता है. यहाँ पे सबसे पहले ये देखो, क्राई टॉक्सिन्स होते हैं, बाइंड होते हैं किसके थ्रू? गट का ल्यूमन माने जहाँ पे इंटेस्टाइनल सेल्स होती हैं, वहाँ पे बाइंड होते हैं. अब जब ये इंटेस्टाइनल सेल्स में बाइंड होगी, तो यहाँ पे स्वेलिंग क्रिएट होगा. स्वेलिंग क्रिएट करने के बाद वो सेल्स डैमेज होंगी. वो सेल्स डैमेज होंगी तो इन्फेक्शन कॉज करेंगी. फिर दूसरी सेल्स भी डैमेज होंगी. फिर इस इंसेक्ट की 2-3 दिन बाद डेथ हो जाती है. अल्टीमेटली डेथ हो जाती है बिकॉज ऑफ द टॉक्सिसिटी क्रिएशन. टॉक्सिन बाइंडिंग कॉजेज द सेल लाइसिस. सेल लाइसिस मीन्स यूं ही सेल लाइसिस, एक ही बार में इंसेक्ट नहीं, डेथ नहीं हो जाती है. सेल लाइसिस होती है, पूरी प्रोसीजर होती है, डिस्ट्रॉइंग होती है. अल्टीमेटली इंसेक्ट क्या होगा? यहाँ पे डेथ हो जाएगी. ठीक है. दिस इज ऑल्सो लिटिल बिट एक्सप्लेन डायग्राम. सो यू कैन सी, यहाँ पे क्या है? कॉटन सेल्स है. कॉटन सेल्स मतलब प्लांट है. ठीक है. कॉटन सेल्स है, प्रमोटर ऑफ द बीटी जीन फ्यूज विद द प्रोडक्शन ऑफ द प्रोटॉक्सिन इन द प्लांट टिश्यू. प्रोटॉक्सिन का फॉर्म में होता है. फिर ये टॉक्सिन फॉर्म में कन्वर्ट हो जाता है। इंसेक्ट ईट द प्लांट टिश्यू, गट एंजाइम्स ऑफ द इंसेक्ट एक्टिवेट द पीटी टॉक्सिन। फिर यहाँ पे जैसे गट में चला जाता है, यहाँ पे गट में क्या होता है, पोर फॉर्मेशन होता है, और ये पोर फॉर्मेशन होगा तो जो सेल्स की इंसेक्ट की जो गट होती है वो रैप्चर हो जाती है और सेल्स की क्या होती है, डेथ हो जाती है। अल्टीमेटली इंसेक्ट भी क्या होगा, डेथ हो जाएगा। ओके, so this is the basically, and there are the lots of the BT strains. ऐसा नहीं है कि आपके पास एक ही दो BT strains है, बैक्टीरिया की भी बहुत सारी स्ट्रेन्स हैं, एंड क्राई के भी बहुत सारे टाइप्स हैं। लाइक आपने पढ़ा होगा क्राई फर्स्ट एवी, क्राई सेकेंड एसी, क्राई थर्ड एसी, ए बी, ऐसे करके लॉट्स ऑफ ऑलमोस्ट 60 टाइप्स हैं। इवन अब मीन्स जो अब जो डिस्कवर्ड हुआ तो 60 से भी ज़्यादा होंगे। Different PT strains produce the different cry proteins, and there are the hundreds of known strains. Scientists have identified more than 60 types of the cry proteins that affect a wide variety of the insect. Most cry proteins are active against the specific group of the insects, such as the larvae of the certain kinds of the flies, beetles, and moths. For example, Colorado potato beetle larvae are affected by the cry3 proteins. Cry1AC is used against the tobacco worms, and European corn borers can be killed with the Cry1AB, Cry1F, Cry1AC, and Cry9C proteins. Other cry proteins are active against the mosquito larvae, flies, or even different types of the nematodes. So these are the varieties you can see here. There are the lots of the varieties of the cry proteins also, and lots of varieties of the BT strains also, means bacteria strains are also there. You can use in the gene cloning as well as the tissue culture methods. Then you can prepare the BT cotton, BT brinjal, BT soybean. Lots of the crops are also there. Now, what is the basically structure of the cry protein? So here you can see there are the cry proteins in that three domains were there. The cry protein has the three domains: an alpha helical N-terminal domain, domain first, a Greek key beta seed domain second, a jelly roll antiparallel beta sandwich domain third. You can see here this is the alpha helical. Okay, and second one is that is beta seed. First one is the alpha helical. It is also very important for the exams. First one is the, there are the three domains found in the cry proteins. First one is the alpha helical. Second one is the beta seed, and third one is the, you can see, antiparallel B domain. Okay, and this three domains are conserved in all types of the cry protein. Little bit structure can be vary, like in that components are there, like cry first AV, some components different, but domains will be fixed on that. The core of the proteins is the most highly conserved, with the helix 5 of the domain first, and the areas of the contact between the domains being the most highly conserved. Okay. Now, the preservation of the BT technology is critical because means ये ऐसा बोलना चाह रहे हैं कि जो BT टेक्नोलॉजी है ना उसका प्रिजर्वेशन थोड़ा डिफिकल्ट है. Why it is difficult? Bollworms, tobacco worms, pink bollworms continue to develop resistance to foliar applied insecticides. Foliar applied insecticides. Because when you are going to continuously apply insecticides, the insects will be resistant for that. Use of the insecticides is tied to ecological concerns. And yes, when you are using the insecticides, then it should not be harmful for the ecological environmental also. Because when you are going to apply, just like a fertilizer, if you are going to apply the fertilizer in crop more and more, then what will happen? Plant is going to resistance. I think you know the best example of glyphosate, why it is banned now. BT genes are valuable resources. Definitely BT genes are valuable resources. You cannot use anytime because it is also harmful to the human beings also. This publication focuses on how BT cotton affects the cotton bud worm as well as the tobacco bollworm also. These are used for the tobacco also. What are the benefits and what are the drawbacks? You can see here. Very if it's able to withstand pest attacks, means if you will compare the BT cotton and normal cotton crop, so what will happen? What will be the benefit? That is, it can stand with the pest attack. Means BT cotton stand with the pest attack. Minimal insecticide and fertilizer required, lowering production cost. When you are using the directly gene, so definitely insecticide you are going to not prefer that, and you are not going to prefer fertilizer also, because there is no need that much. That much means whatever you are using for the normal BT crop. Increase the productivity in the smaller and land patches. Definitely, if you are going to save your crop, productivity will increase. Profit well for the marginal farmers, definitely, because farmers cannot buy the insecticide and fertilizers in varieties of those. So gene get resistance means everything will be fine. Drawbacks of the BT crop: seeds more expensive than the conventional and common variants. Definitely, if you will compare the normal cotton crop seed and BT cotton seeds, it will be very expensive as compared to normal crop, because the efforts has been already done for that. Pest may grow immune to the GM seeds. Natural gene flow between the crops is fatally sidelines, means whatever the natural genes was occurring now, it is not going to be occurred. Ethical issues: definitely we are disturbing the plant also, we are disturbing the bacteria also, and because we are human and we are selfish, so definitely we are doing the harm, we are doing the harm to the bacteria as well as the plants. We are disturbing their genomes also, means, so ethical issues always will be there. Some other BT crops examples like BT brinjal. You have to remember that is cry first AC. BT brinjal is also produced by genetic transformation of the crystal protein gene cry first AC from the bacterium Bacillus thuringiensis. And there are the lots of the crops like a BT soybean also, but they are not approved in India. So this was all about the BT cotton crop. And whatever your queries, you can comment on the comment box. I will try to give answer. And thank you so much, all of you. Thank you so much.